The brain that refused to die. The brain died. The heart stopped. The blood stopped. The oxygen stopped. And the brain stopped. 4 hours. 4 full hours. No movement, no signal, no life. Then, it started again. We're not telling you a science fiction story. Not Frankenstein. Not a movie. This actually happened. In a real lab. At a real university. Published in the most prestigious scientific journal on the planet. What you're about to learn will change the way you think about death. Not give you answers. No. It will make you ask questions you were never supposed to ask. Is death a moment? Or a process? Is there a difference between a dead brain and a brain that is dying? And most importantly, what happens if we could stop that process halfway through? The facts, by the numbers. Right now, 4 hours. The time the brain had gone without any oxygen before the experiment began. 32 pig brains. The number of brains used by researchers in the core experiment. 300 pig heads. The total used by the research team over 10 full years while developing the device. 6 hours. The total duration of the experiment after connecting the brain to the machine. 10 hours total. From the moment of death to the final observations, and the cells were still active. 0. The number of brains that reached consciousness. And that was the plan from the start. 1. The number of emotions you'll feel when you learn that neurons fired again. What exactly did they see? Neurons reassembled. Myelin, the insulating sheath around nerve fibers, reformed completely, just as it was before. Mitochondria, the cell's power stations recovered. Tiny blood vessels reopened. The synthetic fluid began to flow. The brain started consuming glucose, absorbing oxygen, expelling carbon dioxide. The brain's immune cells, called glial cells, activated and began fighting inflammation as if they were inside a living body. And more than that, small slices of brain tissue were taken for testing, and the neurons inside were capable of generating electrical signals. They fired again, briefly, but they fired. We need to go back a little. New Haven, Connecticut, America, an ordinary city, a far from ordinary university, Yale University. Inside the university hospital, there was a lab that had been running for years. A lab working on a question that seemed simple at first. What exactly happens to the brain after death? Not death as a philosophical concept. No. Death meaning the stopping of blood, the stopping of oxygen, the stopping of the chemistry that makes a brain a brain. Dr. Nenad Sestan, a neuroscientist of Croatian descent who studied at Yale and became a professor, there was working on small brain tissue samples taken from animals hours after their death. And he noticed something strange. Those tissues hadn't all died. Some cells were still behaving as if they were alive, even after the entire body had gone. Could this be something? That question took 10 years of work to pursue. April. The research team woke up one morning and drove to a slaughterhouse near New Haven. They brought back 32 pig heads. The pigs had already been slaughtered for food. The team didn't kill anything for the experiment. They returned to the lab, carefully extracted the brains from the skulls, placed them in a dedicated chamber. They connected the major blood vessels, the primary arteries, to a large machine. A machine that works like a heart. It pumps, it pushes, it circulates. But it wasn't pumping blood. It was pumping a special solution. A solution containing synthetic hemoglobin, the oxygen-carrying protein. But without red blood cells. It contained anticoagulants. It contained cell-protective compounds. And it contained something else. Something important. We'll come back to that. The pumping began. 4 hours after death, the lab fell silent. Every eye on the monitors. Then they started seeing what they never expected. The brain turned pink again. The brain that had been gray because there was no blood in it began to regain its color. The microcirculation inside the brain reactivated. The cells began to feed. The neurons began to function. And most critically, the brain began producing carbon dioxide. That means it was breathing. That means metabolism was happening. But no consciousness, no movement, no organized brain signals. Where's the line that separates this from life? This is where the question came that changed everything. If they hadn't added a compound to the solution, one that blocked neurons from fully activating, would those brains have felt something? Nobody knows. The team said they didn't want to find out. Medical equipment was standing by anesthesia and cooling in case any brain showed any sign of awareness. The team was more afraid of the answer to that question than anything else. Let's understand what actually happened. From a scientific standpoint, when the heart stops, blood stops moving. Blood that carries oxygen. The brain, the organ most sensitive to oxygen deprivation in the entire body, begins to deteriorate within minutes. The accepted medical consensus for decades was 4 to 6 minutes without oxygen equals irreversible brain cell death. That's what they taught in medical schools. That's what the definition of brain death was built on. BrainEx shattered that assumption. The brains were not permanently dead after 4 hours. They were in an intermediate state, a state we had no name for. Not life, not complete death. Something in between. How? Because cellular death is a process, not a moment. When oxygen stops, cells don't die instantly. They enter a cascade of reactions. Acids accumulate, membranes break down, energy drains away gradually. BrainEx interrupted that cascade in the middle. The synthetic solution bought time. It supplied cells with energy, stopped the inflammation, reopened the vessels, and restored the cell's chemistry back to its normal course. The result? Cells recovered. Not all of them, but many. 3 years later, same team, same university. But this time, not just a brain, the whole body. They brought in pigs, artificially stopped their hearts, and after a full hour of cardiac death, they connected them to a new device called OrganX, the larger evolution of BrainEx. And what they found? The heart fired again. Electrical signals began to generate inside it. It began to contract. The liver, the kidneys, the vascular system, all of them began recovering at the cellular level. But what stunned the team was something else entirely. The pigs kept under full sedation throughout. Some of them made involuntary muscular movements in the neck and head. They weren't conscious, not alive in the full sense of the word. But their bodies moved. Dr. Sistan said they had no complete explanation for those movements, but they suggested that certain functions of the motor system had been preserved. Dr. Christoph Koch, director of the Allen Institute for Brain Science, who wasn't part of the experiment and was seeing the results from the outside, said it was the most astonishing thing he had ever witnessed in his life as a neuroscientist. He wasn't alone. The research paper was published in Nature, the most prestigious scientific journal in the world, alongside a commentary from 3 bioethicists, stating that the discovery shakes the foundational assumptions about what it means to be alive, in animals and in humans. So why does this matter? Why could what happened in a small lab in New Haven change the world? First, brain death and organ donation. In the United States alone, more than 100,000 people are on the organ transplant waiting list. Every day, 17 people die waiting for an organ. Most available organs come from people declared brain dead. If BrainEx or OrganX can restart brain cells after death, what exactly is the definition of brain death? If cells can be brought back after 4 hours, were they actually dead? And who decides? This isn't just a medical question, it's a legal question, a religious question, a human question. Second, stroke. Every year, 15 million people around the world suffer a stroke. In a stroke, blood supply to part of the brain is cut off, and cells begin to die. Currently, we have an extremely narrow treatment window. 3 to 4.5 hours from the onset of symptoms. After that, the damage becomes permanent. BrainEx suggests the damage doesn't have to be permanent. Cells can recover if given the right conditions in time. If this were developed for humans, not soon, but if it could extend that window, it could bring stroke survivors back with far less damage. Third, brain research itself. Neuroscience has a problem. The living human brain is extremely difficult to study. Tissue sources are limited. Neurological diseases like Alzheimer's and Parkinson's don't have an ideal research model. BrainEx offers a new model. A large animal brain, not a mouse. A brain far closer to the human brain in size that can be studied after death while fully preserving its shape and architecture. The team itself, Sestan and his colleagues, founded a company called Bexorg, working to make human brains from donors available for testing drugs for brain diseases. Not science fiction. It's happening now. Fourth, the definition of death itself. For more than a century, medicine had a simple equation. Heart stops equals blood stops equals brain. Dies equals death. The law was built on it. Religion responded to it. Philosophy debated it. BrainEx says, the equation isn't always right. What does this mean legally if we discover that people declared dead were still in an intermediate state? What does this mean philosophically about the nature of consciousness, soul, and identity? Who is responsible for the decision that someone is gone? Professor Jonathan Moreno from the University of Pennsylvania, the bioethicist, who read the research paper, said that if any subject needed deep public conversation about the ethics of science and medicine, this is it. He didn't say it out of excitement. He said it out of fear. Literally. We're not going to tell you what this means. That's not our job. But let me ask you one last question. If the brain died, and if we could bring it back, who exactly came back? Are the memories still there? Is the personality still intact? Is the one who knows itself as you still in there? The brain can recover at the cellular level. The cells can function. The neurons can fire. But the cells are not you. The electricity is not you. The scientists asked, when does the brain actually die? Our question is, when does the brain become itself?